So in this last video, we're going to be talking about recycling material to the lysosome. And I want to tell you that the surface of the cell is a dynamic place. And that's because there's always going to be material exiting the cell. And so there's going to be exocytosis of secreted proteins. And depending on the cell type, this could be happening all the time or infrequently. There's also proteins that age on the surface, and they often need to be recycled, so taken off of the surface. And the lysosome is the place to do that, to break down these large macromolecules. Some cell types, like the cells I study, called phagocytes, ingest material that's pretty big, like pathogens or dying cells. And the reason they ingest it is so that they can degrade it, and this degradation happens in the lysosome. So what we're going to be talking about is how material gets to the lysosome. So material is either brought in one of these three ways. So material can be brought in by pinching off of the plasma membrane. And this can occur when small things are taken up through endocytosis, and when large things are taken up, specifically something greater than 0.5 microns, and that's called phagocytosis. And there can also be ways to remove damaged organelles. In this case, these organelles are surrounded by a new membrane that forms around that damaged organelle, and then they're fused with the lysosome, and this process is called autophagy. These are just a few pathways that exist that bring material for the lysosome, so these are the ones we're talking about today, but there are more. So the first one we're going to talk about is receptor-mediated endocytosis. And so there's two ways endocytosis can occur. One of them is when a receptor recognizes a ligand, which is a molecule that binds a receptor. And the other one is just taking up the ligands without a receptor around. So right here, we're looking at in blue is our receptor, and it's bound its ligand. And what happens is you see is that there's this inward budding of the plasma membrane, and then this bud pinches off. And when this happens, what forms is called an endocytic vesicle. And so there's been a little bit of a membrane contraction here, and usually requires some sort of pinching to occur. So now we have our endocytic vesicle and our receptor still bound to our ligand. The next step is that this vesicle traffics on the cytoskeleton, and it arrives at this early endosome. Okay, now this endocytic vesicle delivers both its contents and its membrane. So I'm showing this kind of black part of here fusing with this early endosome. And this early endosome has a relatively lower pH than does the cytosol or the extracellular space. And at this relatively low pH, or lower pH, the receptor and the ligand lose affinity for one another. And so two things can happen here in this early endosome. One is that part of this early endosome can bud off again, and it can recycle this receptor to the surface of the cell. So that's one thing that can happen, and this is called receptor-mediated recycling. But the other thing that can happen is that this material then continues to move on in the pathway. And so it moves along microtubule tracks to become a late endosome. And so what you'll notice here is that the pH has dropped more. And so what's happening is there's now usually a pump on the surface that gets added by other vesicles that are departing from the Golgi are now fusing with this, and it's picking up a pump that can pump protons, and it's picking up acidic hydrolases. And as these vesicles fuse to make this more acidic, this compartment grows in size. This material then fuses with the lysosome, and the lysosome it would become bigger, except it can also break down membrane. And the lysosome has a pH of around 4.5. And this is usually where any acidic hydrolases that have been dropped off from the Golgi, this is the pH they're most active. And so this material here, then this ligand becomes degraded pretty fast once it reaches that lysosome. this, let's say this little triangle here represents a protein, then those peptide bonds would be broken, and then those amino acids could be broken down, or they could be used in another process. So this is the overall process of receptor-mediated endocytosis. I presented this to you in a little bit more detail than what's in your book, but it's shown again here. Here we have our endocytic vesicle, and then it's fusing with this early endosome that becomes the late endosome, which then fuses with the lysosome. And then at this early endosome phase, you can also get recycling of the receptors back to the surface. And the reason I presented it in just a little more detail was that I think that there's just a few too many things missing from this figure in the textbook that makes it seem like this vesicle is just changing randomly. And so I wanted to provide a little bit more context. But once it reaches the lysosome, this material gets degraded. The other process is called phagocytosis, and this is one thing my lab is very interested in. And in this case, the membrane either, again, this figure is really oversimplified, the membrane either sinks in with the particle, and then that forms this compartment, this new compartments form once it pinches off, and that compartment's called the phagosome. And this phagosome is then trafficked, again, using motor proteins and cytoskeletal filaments to the lysosome. And when it empties its contents, it can destroy that content. And if this is a pathogen, it can try to and hopefully kill that pathogen. There's also a way that the membrane can kind of come up and zip around the particle before it comes in, but this is just a very simplified diagram. And then the last pathway that I said I would talk about is called autophagy. And so sometimes an organelle, like the mitochondria in this example, can become damaged. And this damage isn't good for the cell because you can start to leak out things that shouldn't belong in the cytosol. And so what happens is that there are cues sent out to proteins in the cytosol, and then those proteins begin bringing bits of membrane to this damaged organelle, and they start surrounding it until it's fully encased. Once it's fully encased, we have a new organelle that forms, and we call it the autophagosome. Auto because it's surrounding self, and then this autophagosome picks up signals to direct this newly formed organelle to the lysosome and break up this damaged organelle inside. So these are the three pathways that bring recycled material to the lysosome. So again, material is trafficked into cells through endocytosis and phagocytosis. These distinctions depend on size, and this material ends up in the lysosome via organelle trafficking. And then another way material can get to the lysosome is when there are damaged organelles and autophagy is induced, and this will cause an envelope to form around those membranes so that they can be trafficked to the lysosome for destruction. So that's it for today's videos, and I look forward to seeing everyone in class. Thanks, bye.